Hello everyone, my name is Dr. S.K.M. Habeeb. I also go by the name Habeeb Sheikh Mohidin. I'm working as an associate professor in the Department of Genetic Engineering at the SRM Institute of Science and Technology located in Chennai. I have come up with a concept to explain to you people the basics behind the sequence alignment and also the different methods that are available which could be used to perform this sequence alignment. Let's move on and have a look at these concepts now. What is sequence alignment? Now let's assume it's like you have two different sequences and these sequences are of different lengths. And these sequences also consist of different types of characters. So what we try to do here is align them under each other in such a way so that we could extract important features, signatures, or patterns that could hold very significant message. Now this message could be a domain which talks about the functional importance of a protein or a gene. It could be a motif region, and that could talk about the structural aspects about the protein. So this is the purpose of aligning sequences. So sequence alignment further can be divided into two different types. It can be called as a local alignment and a global alignment. Now I would like to make it much more easier for you people by saying that if you want to align the sequences from the very first character till the end so that all the positions in those two sequences are mapped, that is called as a sequence alignment, called global one. Whereas if you are giving importance to only the localized regions which could preferably hold functional or structural significance between the two sequences, so therefore the alignment only gets concentrated upon that specific region. So such an alignment is termed as a local alignment. Now in a broader context, sequence alignment can also be categorized in another way. If the alignment involves only two sequences, we call it as a pairwise sequence alignment, or if there are more than three sequences involved in the alignment, we call it as a multiple sequence alignment. What these alignments can help us with, as I just stated a short while ago, they can help us in understanding the similarity between the sequences. Now, if two sequences are similar enough, that tells us that they are homologous, which means these sequences have derived from a common ancestor. Now, that brings us to a further concept to discuss about what is a conserved and what is a diverge. A conserved region is a region that is common between the two sequences, which has been simply evolved from its common ancestor. So let's move on and have a look at this particular page. Now you can see there are two sequences in the first example under global alignment. And these two sequences, if could be seen, though they are of equal length, but when they are arranged under each other in such a way, you can see there are so many mismatches or non-matching characters placed under each other. Whereas once you perform the alignment, you can see the length of the sequence, yes, absolutely has increased by 2 positions. However, the number of matching regions or matching characters that have been found to be placed under each other has increased. Now, this is the beauty of sequence alignment. Now, these dashes, which are nothing but they signify the insertions or deletions. So on the top, the global alignment, the alignment as could be seen starts from the very first character and ends till the end. Whereas a closer look at the image on the bottom part of the slide gives you guys an idea. No matter there are two sequences are of equal or unequal lengths, but the emphasis is only on a concentrated region, and therefore the alignment is only concentrated on the specific region where more number of matching regions are witnessed. In this case, we aren't bothered about the entire stretch, but we are bothered about only fetching the regions which are highly similar between the two sequences or more sequences. So this is the basic difference between a global alignment and a local alignment. Now let's have an example. Now what you see over here, it's a snapshot of a blast. What are you looking at over here is just an example for you people to understand how alignment is performed. So, what is an homology, similarity, and identity? Now, in simple terms, identity can be considered to be as you have a character, say A, that stands for an alanine amino acid, and in the next sequence with the same position must also consist of alanine strictly. So this is called identity. In other words, I could say that identity can be related to as looking at yourself in the mirror. So there shouldn't be any deviation, but the similar character must be present in the position in the two sequences. So that defines the identity. Whereas similarity, as you guys know, that the amino acids can be classified depending upon their different physicochemical characteristics. So if two amino acids in a particular position in two different sequences are found to be having similar physicochemical properties, say for example alanine placed under valine, obviously these are aliphatic amino acids and therefore they are called as similar amino acids. Now what is homology? Homology is two sequences that are shared from the same common ancestor are called as homologous sequences. Therefore, we can move on to the next slide. So what are the different types of homologies do we have? Now homologous are the genes that have been derived from a common ancestor. Perhaps there are different types of further homologous categories. Say for example, the orthologous and the paralogous. Now, the orthologous are the genes which are derived from the common ancestor but have evolved due to speciation and are found to be present in different species. That's what you call as an orthologous. Whereas paralogous are the homologous sequences that have evolved due to gene duplication, therefore they are present within the same species. A best example, myoglobin and an hemoglobin in the same species. So therefore, two types of homologous sequences, that is orthologs and paralogs. Now you can look at this particular image on the left hand side at the bottom part of this particular slide. A is the common ancestor. Now from A you have B and C. These are the two species that have evolved. So when they evolved, they inherit all the functions from A. So therefore B and C become the homologous sequences because they are derived from the common ancestor. In this case, this happens to be the species A. Whereas a gene duplication, in this case C to C dash, is nothing but the gene has been duplicated but would perform a similar but different function within the same species. So therefore, there is no speciation that is going to take place in paralogous, but there is gene duplication that is going to take place. And this is the reason I gave you people an example of dealing with human alpha globin and human beta globin over here. So therefore, B and C are termed as orthologs, whereas C and C dash are termed as paralogs. I hope that has given you guys a good idea to get an understanding about the homology. So here, how is this sequence alignment performed? Is there an approach? Yes, we do. There are multiple ways the sequence alignment could be achieved. There are simpler ones, there are tedious ones, and there are, the sophisticated ones. We call it as the heuristic methods. So the one that we are going to discuss in this particular session is the dot matrix method, which is also called as a dot plot. The another one is a dynamic programming algorithm. It's a very, very rigorous and time consuming one, but the most accurate to be on the planet when it comes to sequence alignment. And then you got the word method, which is also called as a k-tuple method. So for the purpose of this particular session, we will move on to have a discussion on the dot plot method. Back in 1970s, it was a duo of Mr. Gibson, Mr. McIntyre. The two gentlemen came up with a concept called dot matrix. It's not a difficult method to understand. I would rather put it up like this. We generally plot the graphs on multiple occasions, where you got an x-axis and you got a y-axis. So it goes like this, and you got a unit per cell where you place one character per unit per cell. So how actually does this work? Let's get into the concept. Imagine you got two sequences, sequence A and sequence B. We write one sequence across the page on the top, and we write another sequence down the page from the top to bottom. This is done in such a way every cell occupies one character. And then you move on to find the matching cells in this. This would give you guys a very good idea about what actually a dot plot is. So now one sequence is written across the page and one sequence is written down the page, and now you will place these bases or amino acids in such a way that one cell occupies one character. Further, you search and scan across this matrix to find the regions where you have identical characters, and therefore if you find one, you would place a dot. So in this case, a G to G match is recognized with the help of this dot. Likewise, these types of dots are placed in the intersection cells wherever the two characters in the X and Y axis or A and B sequences are matching. So this gives us an array of multiple dots, and this is why we call this method as a dot plot method. So this is a graphically easier method because the next step in dot plot is to concentrate and look out for the regions which has the longest stretch, kind of a diagonal. So you look for diagonals, you connect these dots in search of finding the longest possible diagonal. Longer the diagonal, longer the length of the diagonal, it means more similar these two sequences are to each other. So this is the basic concept of dot plot. As I said, dot plot method is a very interesting method. It's the easiest of the method. Why? Because it gives you guys a clear understanding about the similarities between two sequences that you are going to analyze over here. However, dot plot method will not give you guys a residue based alignment as could be obtained from other methods. Please remember, dot plot method is a graphical representation method which graphically helps us in getting an understanding about the similarities between the two sequences. It's a two-dimensional graph. I should put it up like that. So now the next image that you are seeing right at the bottom is, you can see there are multiple dots. Now what you are having over here, it's a window of 200 characters to 200 characters, and it's kind of a very noisy plot. Imagine you take up a whole gene running into thousands of bases. Imagine you constructing the dot plot for an entire chromosome versus chromosome. That becomes trickier and that becomes messy. So what do you do under such a circumstances? Now what you see over here, there are two important terms. One is a window size and the next one is a stringency. In this case, the window size is 1 and stringency is 1 as well. What does that mean? A window size is nothing but the number of characters that are to be placed or accommodated per cell, whereas stringency is when to place a dot. So in this case, if the stringency and window size are 1, so it means that there must be 100% match in the intersection region. Only then a dot is placed. Otherwise, we don't place a dot. So what is the drawback of having one as a window size and a stringency? The drawback is you will have an image that is there on the right hand side on top, something looking like this, and therefore it becomes very trickier to draw a diagonal. So therefore what happens is you come up with a different concept, a further better idea, where you select a larger window size and you also come up with a stringency value. Now what is that? Instead of placing one character per cell, set of contiguous characters could be placed in such a way in one cell. Therefore, then you decide what is the criteria for you to place a dot, for you to consider that section to be similar. In this case, in this example, the stringency used here is 7. Now what do you mean by that? When position X and position Y are mapped, and if 7 out of 23 characters are matching, only then we are going to place a dot. Otherwise, a dot is not going to be placed. So this is how you reduce the number of dots that are going to be placed. And this is how noise is reduced. The image on the top and the image right there at the bottom of it gives you guys a clear idea about the amount of noise that is present in this particular dot plot on the top, whereas the one right at the bottom for the same sequences gives you people an idea about how we have reduced the dots and how it has clearly helping us in getting an understanding about the nature of similarity between these two sequences that we are trying to align. So this is the significance of using appropriate window size and stringency. Generally, the dot matrix analysis can readily reveal the presence of insertions and deletions. You can simply look at the dot plot and you could have easily see where there isn't a possibility of finding a diagonal. There could be a gap right at the center or somewhere, and that region specifically tells us that there is a deletion or there is an insertion in the another sequence. So due to the simplicity of this method is also used for finding, the direct and inverted repeats. However, there are some limitations to this method. What is the best limitation? The dot matrix method, as I just explained to you just a short while ago, it is only going to give us an understanding in a two-dimensional graphical way. It is not going to help us with the actual alignment. So yes, this method can be taken up as a pre-decider to proceed further, whether to check and proceed with rigorous alignment exercise. This is one of the major limitations of the dot matrix method or the dot plot method. Generally, sticking to the concept of window size and window stringency, considering that 1 out of 4 bases is going to be the same, so therefore it is a practice to follow a larger window size for DNA sequences, whereas for protein sequences a very smaller window size is preferred. And also a much lenient approach is followed with respect to the DNA sequences when it comes to the stringency, whereas in the case of proteins it is relatively much strict. As you know, the possibility of finding the same amino acid is 1 in 20 during the alignment. So due to these reasons, the DNA sequences attract different types of window size and window stringency, whereas the protein sequences attract different window size and stringency. So with this, I hope you people have got an idea about what actually a dot plot method is. Well, these are the few textbooks that had been utilized. One of these textbooks or one or more of these textbooks had been utilized to come up with an understanding about this particular topic. I thank all the authors who had come up with such a fantastic textbooks. I thank the SRM Institute of Science and Technology. Now in our next session, we will be looking into the concepts of global alignment using Needleman-Wunsch algorithm. Until then, thank you everyone. Have a good time. Bye-bye.